Good evening, everybody, and welcome to the second virtual NRES Internship Forum, and I would say the first spring rendition of the Internship Forum. So we're glad you're here. Normally during internship forums over the years, we have our mouths full of pizza, and we would also be in the good barn if it wasn't for the pandemic. But I'm glad that we're able to still do this and hear from our students. Some of you have had experiences move forward despite this pandemic, and the nature of the program lends itself being interdisciplinary to leveraging this wide range of experiences, all the way from 395 basic research projects to more applied projects and internship experiences. So we look forward to hearing from you all. Experiential learning has played a central role in the training of NRES students over the 27-year history of the program. I might add that in some cases, these experiences have guided students in their future careers, and whether it be ruling out what they don't want to do or sort of validating what they do want to do. And so it's a very valuable experience. We look forward to the day when we can all meet back face to face. As a matter of sort of housekeeping, in the past, students have wanted hard copies of their posters. Everything's virtual now, but if you, for some reason, want a hard copy of your poster, feel free to contact me, send me an email, and we can get those printed. Before I turn it over to Adia, I'd just like to thank Larry, who has been instrumental in organizing this event, as well as Adia, and even proposing that we have a spring internship forum. And so I really appreciate their time. Without anything further, I'll turn it over to Adia.
Hello, everyone, and welcome to the Internship Forum. So just a few housekeeping notes. Everybody is already on mute. If you could stay that way while our speakers are talking, that would be wonderful so that we aren't interrupting each other. If you have questions, you can either raise your hand or put them in the chat. Each student will have about 5 minutes to present, after which we will have about 2 to 3 minutes for questions. So the best way to do that, if you have a question, is just to wait till the end during the question session to ask them. Before we get going, does anybody have any last-minute technical questions? No, all right, wonderful. Well, our very first presenter is Casey Durham. Casey worked with the Kentucky Geologic Survey on a project studying springs. So Casey, can you please share your screen?
Absolutely. All righty, can everyone see the screen? Okay.
Yes.
Yeah, I'll minimize this. Alrighty, well, good evening, everyone. My name is Casey Durham, and I'm a senior here in the NRES program. Before I start, I really want to thank Dr. Sobey for directing me to Dr. Tobin so that I could do this research project. He really pushed me and challenged me during our remote experience, and he made me find my new interest for GIS. So thank you very much. So what we did during the fall 2020 semester, I partnered with Dr. Tobin from the Kentucky Geological Survey to create my research project. After weeks of background research, I discovered that a lake in my hometown of Bergen, Kentucky, which is an hour south of Lexington, was actually impaired. Harrington Lake has been listed as impaired by the EPA since 2006. It was impaired due to high concentrations of methylmercury in fish tissue. Mercury is a bioaccumulative and toxic chemical that persists in a substantial amount in Harrington Lake. I wondered whether the high values were due to the presence of the E.W. Brown coal-fired power plant, which sits here at the northernmost point of the lake, and they also actually created it. I wanted to know, are the high mercury levels in Harrington Lake caused by a local source? The leading cause of mercury entering waterways in the United States is actually through fossil fuel combustion. Coal received from this power plant is actually from southern Indiana, which has a high sulfur content. mercury is actually bound to sulfur, which is found in coal. So once it's combusted, mercury is released into the atmosphere and then deposited by air or water in the form of rain. This is a really big concern because not only can mercury accumulate in aquatic systems, but in us as well. It absorbs into our bloodstream and it can cause fertility, neurological, cardiovascular health problems. Some of those are linked to ADHD, autism, Tourette's, gastrointestinal irritation, and low birth weights. So what I did is I started to create GIS maps. And so what I did is I collected water data from the Kentucky Watershed Watch program and data for the groundwater from Sarah Arpin at KGS. This data is what is displayed onto the maps that you see here. I use an inverse distance weighting tool, which is also known as IDW spatial analysis tool, to create the following raster image that uses the mercury concentrations from the data points to pretty much improvise and fill in the spots across the landscape. All of the values that you see here in the legend are above the EPA's maximum contaminant level for aquatic systems at 0.0014 milligrams per liter. As you can see, in these areas here, it is substantially over the level that they list. In order to determine the average that was supposed to be for the regional, which would be expected, I had to extract data from the National Atmospheric Deposition Program. This organization collects data from measuring stations across the United States. So for Harrington Lake, we use two sites, the Clifty Falls State Park Measuring Station in Madison, Indiana, and then the one here in Mammoth Cave in Kentucky. I averaged the concentrations for the years of our area of interest for 2006, 2008, 10, 12, and 14. Fortunately, when we did our groundwater data, it's not as frightening as the surface water is. The levels of mercury are beneath the surface. Beneath the surface is still over the EPA's maximum level. However, the groundwater samples do not reflect the intensity at the surface level. The level sampled from Harrington Lake surface water data was around 43,000 times higher than what the National Atmospheric Deposition Program reported. The total maximum daily loads of mercury from the plant surface impoundments and air stacks resulted in concentrations that are astronomically higher than the regional concentrations. These estimates were made by averaging the total maximum daily loads from each of the 5 focus years and dividing that by the number of liters in the lake. Harrington Lake and surrounding area has a concentration of 0.30 milligrams per liter. The differing surface water sample results from KGS and concentration estimates indicate that the heightened levels are in fact from a local source. The total maximum daily load suggests that it may be from the E.W. Brown plant. It's important to note that the map of the concentrations on the higher end of the range are not found in the immediate area of the power plants. They're rather found away from the plants in the northeast direction. And this is because mercury attaches onto particles in the atmosphere and travels to the northeast wind direction that central Kentucky has. Data suggests that a local source is contributing to the high concentrations of mercury in Lake Harrington, but in addition, it is also affecting areas that are away from the plant. Does anyone have any questions?
I have a question. So I have a friend that has a house on Harrington Lake. What should they be concerned? Should they not swim? What's the deal?
One of the biggest things that they warn against is just like if you consume fish out of Harrington Lake. I don't know if she does this, but a lot of people that actually live on the lake don't eat anything from it. But it's just like the EPA has guides on how much you could consume, like for children they could consume less, and adults it just depends. But where there is such a heightened level of mercury found in fish, you should really limit or just completely not eat anything from it at all, because you can experience those side effects short-term and long-term if you're like consuming anything out of it. As far as water, like you can absolutely like absorb that into your bloodstream, but it's mainly through if you're eating, the fish that come or anything aquatic that comes out of it.
Thank you.
Casey, nice job. I have a question.
Yeah.
You mentioned the EPA level, what maximum contaminant level? What is it for mercury again?
It is 0.0014. And that is for the aquatic, that's not for drinking water. For drinking water, it's like 0.002. But that limit is I focused on the aquatic habitat because that's really what this focused on. And that's one of the main reasons it was flagged and impaired. So that's what I went with.
Gotcha.
Mhm.
So this is kind of frightening. I guess I'm looking at these numbers. You had value
Right.
well above that, right? I mean,
Yes, they were very high. And like whenever we went through a paper, and I know Rob was my experience advisor, and even when we were going through my paper, like it really just flags and you're like, wow, why is that so high? But yes,
wow.
they were definitely higher. And the concentrations of the total maximum daily loads were even higher, but that's also just assuming that everything that was released was put into the water.
Right.
Mhm.
Wow. Okay. Thank you.
Yeah, thank you.
Casey, should people know about this?
Absolutely. Yeah, I mean, I definitely think they should. And a lot of people in my area are not, I don't want to say that they're not concerned, but they're just not like environmentally concerned. You kind of have that thing where people don't think that they can be concerned about issues like this just because they're so passionate about, having a coal-fired power plant in our area. But yes, absolutely, they should definitely know about it. Because a lot of people in our area like to come to the lake and fish. So
Yeah, I'd say. So you said you kind of phrased it in a cagey way. You said that the power plant could be the source
right.
of these astronomically high
Mhm.
mercury levels. Just hypothesize for what else could possibly be responsible for that. Is there anything else that could possibly be responsible for that?
Yeah, I mean, we really like whenever I was doing my research project, I definitely don't think there's really too much else that could be, a direct source. Like we do have a couple of factories in Mercer County. They're kind of concentrated in our area. But if you ask me, I would definitely say that that's one of the number one producers. But, in research, I don't have I don't feel like my data is strong enough to be like, it's them for sure. But I definitely think that they're most likely the source.
Yeah, I can't imagine what else it could be. Anyway, good job.
Thank you.
All the way through. And I should know.
All right, wonderful. Thank you, Casey. We're going to move on from Casey right now, but hopefully at the end we'll have more time for questions if people want to keep asking Casey about this really impressive but also deeply disturbing project that she did. Our next presenter is Josie Gogagan. Joe, I'm so sorry if I just butchered your last name. She has been working as an NRES outreach assistant and helping with the NRES newsletter, and she's going to talk to us about the skills that she has developed at that position.
Thank you. I'll go ahead and share my screen here. Oh. Okay. So, hi, I'm Josie. It's Gagan, but so I have been the outreach assistant for the NRS newsletter since the spring of my junior year. Basically, a lot of my responsibilities just include obviously creating the biannual newsletter and then also attending any type of outreach events, which were obviously a bit lesser on my time in the newsletter due to a global pandemic. But beyond that, I got to sit in on a few steering committee meetings as well, which was really interesting just to kind of see that side of the NRS program that a lot of students don't get to see, and definitely be more of a voice of students who don't really get to voice their opinions on a lot of those types of topics. So that was a really awesome experience. And beyond that as well, I got to conduct a lot of interviews with my peers and with faculty and just other UK staff as well. In terms of kind of becoming the outreach assistant, I basically just applied in my junior year. I discovered that I kind of liked writing and just kind of went from there and applied and I got it. So it's been a really awesome experience since then. Since then, I have also produced 3 newsletters, and for the past 2, me and my co-editor have been taking the lead on those. And by working on these, I've gotten to refine a lot of my writing skills and also expand a lot of my environmental knowledge as well beyond just my focus areas within the program. I got to strengthen a lot of my relationships with professors and meet more of my peers and just really find people that shared that same type of passion for environmental policy and awareness, which is what I want to do with my future career as well. So leading up to here, this was my first environmental alert column that is typically a staple in the newsletter. We typically try to do one every issue. So for last spring, we did COVID-19 waste and basically the effects of gloves and masks and any type of PPE wear, and then any other trash that had been coming from restaurants that were doing more to-go type things, as well as like the dining halls on campus. And this was a really interesting piece to do because it was the first time that I got to express any type of or publish any type of informative or persuasive type of writing. So it felt like a bigger deal to me, and I got to meet some more like recycling coordinators of campus and other faculty on campus in those areas. So that was just very interesting for me to do, and it allowed me to kind of figure out how to write things in a way that could be perceived well by people and taken in a way that people could understand that had no background in types of environmental sciences, but also still be enjoyed by people with an environmental background. So that was a very good first experience for me to have since I wanted to go into environmental consulting, and more of that persuasive writing would be relevant. Along with refining my writing skills and just getting to know more of my peers, I also got to learn more about a lot of Adobe software. So over here, it's a lot of the, we create our newsletter in Adobe InDesign, and we also use Illustrator for any type of our graphics here. So here is just like a snippet of what InDesign is kind of like. I had never done anything with InDesign leading up to this, so it was definitely a big learning experience for me. And now it's a new skill that I can bring with me as well. So I've just been able to refine all my skills in these types of programs. And then also when I got to conduct these type of interviews, I got to gain interview skills rather than being interviewed. I got to now interview peers and people that I looked up to. So I thought that was a great experience for myself as well. And I got to learn about new job opportunities in fields that I probably wouldn't have been super aware of if I had not been in this position as well. And some of those fields include like landscape architecture, food waste, and sustainability, energy resources, and many, many more. And also through this, I've gotten to learn more about the NRES program as a whole, and just as myself as a student, and really come to know what I enjoy studying about here. So. Any questions?
Great job, Joe. I've got a question, self-serving. What would you say to students that are considering applying to be an outreach assistant? What is the best part of the job?
Definitely do it, because I didn't think that it was going to be something that I enjoyed. And like I said, I think the best thing about it has been the part that I've got to like meet all these new amazing faculty and really strengthen these relationships with professors and peers that I definitely would not have done if I wasn't in this position. And I think that's just important in your experience as a student. And I think that's an important experience just to have in life as well, to just meet all these new people and really learn about all these subjects that I wouldn't have gotten to otherwise.
So I have a quick question. Josie, you talked almost entirely about the newsletter, but have you and Julia also been updating social media and engaging with the program in that way?
Yeah, so we also do have our Facebook page that we like to keep updated. And then we sent out surveys as well for a part of the newsletter that's coming out this semester. We sent out a survey of kind of what people would like to see within the program and what they want to see more of, or what they think the newsletter is lacking in itself, and any type of like representation as well. So that is something that we are still definitely working towards, and hopefully this next newsletter will kind of meet some of those people's needs as well.
All right, wonderful. Okay, so we are going to move on. Oh, sorry. Great job, Joe. We're going to move on to Joshua L. Joshua worked with the UK Department of Plant and Soil Sciences. Joshua, will you share your screen, please?
Yeah, sure thing. All right, let's see. Can everybody see that? All right, cool. So I'm Josh. My name is Josh, and I'm a senior in the Natural Resources and Environmental Science program. I'll be graduating this May. It's very exciting. And for my internship stuff that I did, I ended up doing an NRE 395, which is research, pretty similar to what Casey ended up doing. And my project was called No-Till Agriculture and Its Effects on Soil Moisture Retention in a Long-Term Experimental Cornfield. I worked with Dr. Hannah Poffenbarger, and in conjunction with Dr. Ole Windroth in the Plant and Soil Science Department here at UK. And it's all about looking at the way that no-till agriculture affects corn and production. And we're specifically looking at the way that it affects soil moisture retention, so, which is very important, especially in the early stages of growth, because plants need water. So let's see, I'll get this zoomed. This is a quick map of what the tillage study looked like, where it took place. It was on the University of Kentucky Spindletop Farm on a 50-year-old research field known as the Blevins long-term tillage plot. And it's split into 4 different repetitions with different rates of nitrogen applied. For this study, we were looking specifically at fields that had 150 pounds of nitrogen, and also fields that had no nitrogen applied. So it was a total of 4 data sets. And so I'll give a quick little bit of background on no-till agriculture. It's been rising in popularity the last, probably since the 70s and 80s. And the idea behind it is you don't disturb the soil in any way when you plant. All you do is you dig like a little 3-inch trench, and you drop a soil or a seed into it, cover it, and then move on. And that's as opposed to conventionally tilling something, which used to be a moldboard steel plow, where you basically tear up the entire 10-inch substructure of the soil in order to plant and turn over soil. Now that looks more like chisel tillage, which is less invasive, but still more invasive than no-till. So like I said, we were looking at how this is going to affect soil moisture, specifically in the crop rooting zone. So that's about 60 centimeters below the surface of the soil. And yeah, the major differences we were looking between are no-till with 150 pounds of nitrogen, no-till with 0 nitrogen, conventionally tilled with 0 nitrogen, and conventionally tilled with 150 pounds of nitrogen. So this is what we found, and it indicates that there's a statistically significant difference at all levels in the difference between soil moisture retention the difference between conventionally tilled and let's see. Yeah, conventionally tilled and no-tilled fields, regardless of nitrogen application. So it's very important there. And we also found that there's a statistically significant difference in the amount of grain produced by fields that had no-till and fields that were conventionally tilled. So it looks like no-till is on average better in every regard, even with or without nitrogen. So we had, I think it was a total of 26% more grain between the no-till and the conventionally tilled plots, with the biggest impact being in the area that didn't have any nitrogen applied. I think it's almost 85% difference there in terms of get a lot more grain out of the no-till plots. So yeah, after analyzing our data, that's what we found. And let's see, we think that a lot of the reason why no-till fields have this much higher ability to retain soil moisture is because you're just not breaking up the soil structure at all. And you're also keeping any crop residue that you might have lost through either erosion or just turning over in a conventionally tilled plot, which prevents a lot of evaporation. So in the long term, your plant is going to have better access and longer access to water at all of the important growth stages. So, and you also, you're protecting your pore space too, so you can hold more water in the long term. And yeah, that's about it.
Nice job, Josh. I have a question.
Yeah, what's up?
So it didn't look like there was much of an effect on water content comparing the two nitrogen rates. Would you have expected there to be a difference between those? And or maybe not. I mean, I would assume there's more soil organic carbon and a higher nitrogen rate, right? Which might hold more water, but maybe.
Yeah, that's a good question. You honestly, thinking about that, you'd expect to see that the higher nitrogen rates would use more water, so maybe it would be lower in terms of retention because you know they're growing faster. And that's something that we see down here at the 30 centimeter level. This initial dip right there, we hypothesized that the reason that the conventionally tilled plot there had more water in it was because, as opposed to the no-till plot, was because the no-till was just growing faster because it had a better access to water.
Right.
So I think that, does that answer that?
Yeah, I guess just at the surface, it was kind of interesting. Closer up, higher up at the surface, I would expect where you'd see the stratification maybe of carbon there. You might see more water as well, but it didn't look like you saw that. If you go up top, see?
Yeah.
Anyway, I was just curious. It kind of struck my curiosity there. But yeah.
I have a question.
Yeah.
Do you think these results apply to like a small home garden plot, or does this really need to be kind of on a farm scale before you see these sorts of differences?
I think you absolutely could apply this to a garden. It means way less work for you too. So you don't have to, go, if you've got a raised bed or something, go till that up. You don't have to do any of that now. You just plant and I guess maybe fertilize if you want, and you just kind of leave it, make sure it gets water.
Great, thank you.
Wonderful. Great job, Josh. Okay, so the next speaker is going to be Shay Davis. Shay spent her internship with the Kentucky Wildlife Center. Shay, can you share your screen?
Yes, let me pull it. Okay. All right. Okay, can everyone see this? And also hear me? Okay. So, hello everybody, my name is Shay Davis, and I've had the opportunity to intern with the Kentucky Wildlife Center, a place in which I'm sure some of you are somewhat familiar with, as a few NRES students have interned here in the past. With COVID interrupting my initial internship plans, I had visited the center previously and decided it would be a chance to broaden myself in terms of knowledge and hands-on experience. I took on my current position with the center in January to complete as a spring semester internship, and I'm currently still there. And I've been pleasantly surprised with my experience at the center thus far. Kentucky Wildlife Center specializes in wildlife rehabilitation, and it began extremely small in 2006. moved to its current location in 2019 on Georgetown Road, which has allowed the practice to flourish and become a hub for effective wildlife raising, rehabilitation, public education, and conducting research. A full day at the Kentucky Wildlife Center for me starts at around 9 a.m., which consists of preparing specified meals for all animals currently housed on the property, as well as any medications or supplements necessary and any daily required maintenance of their enclosures to ensure their health and comfortability. These animals include about 25 education species and can range from about 30 to 40 animals day to day, just depending on weekly intakes. And those are generally and hopefully quick releases. And what this number doesn't include are injured or orphaned infant animals, which require much more frequent and meticulous care. Once those more time-consuming and pertinent tasks have been completed, there are many other tasks to upkeep facility on the daily, which isn't as exciting, so I won't go into that. But the next focus of each day is preparing for our daily intakes. And so the Kentucky Wildlife Center, on average, intakes over 1,000 individual animals per year, some of which require little care and are ready for release fairly quickly, some for an intermediate stay for injury, medication, surgery, and some that require months before their subsequent release, which that typically includes those infant species that come in. Every animal that Kentucky Wildlife Center intakes must be brought to us. We cannot trap or contain species per Kentucky Fish and Wildlife regulations. Once the intake is received, the person that found and brought the animal must provide information on a form. And once that information is received, the animal is assessed, triaged, and everything is documented no matter what and kept on file forever, even if the animal must be euthanized. I've been able to go through the triaging process independently, obviously still with the supervision of a licensed wildlife rehabilitator on staff. Every detail that can be observed based on the animal's physical and internal condition is written down, and every medication, fluid, or sedative that is administered is also accounted for and put on file with that individual intakes form, and that animal is given a unique ID number. Unfortunately, there are cases where the call has to be made to euthanize an animal based on presence of a highly contagious disease, such as distemper in raccoons, or just injury beyond repair, or any severe neurological damage. And sadly, I have had to participate in the euthanization of some animals. It's very sad. But beyond the basic duties and experiences that every intern engages in at Kentucky Wildlife Center, we have the opportunity to choose a secondary experience that pertains more closely to a specific interest of ours. And so upon my acceptance as an intern, I chose their pollinator pathway, in which I'm currently involved in the propagation of pollinator-friendly species, which will soon be planted in Kentucky Wildlife Center's registered monarch way station. And those species are listed in the middle of my poster there. And then obviously other flowering plants, but the milkweed are most appealing to monarch butterflies, but attract and benefit a variety of other pollinators. And pollinators play such an extreme role in essentially allowing the world to turn, to put it in hyperbolic terms. They allow our food and agriculture industry to flourish, along with many other ecological benefits. We're all fairly well aware of this, especially with the prominent bee issues that have really been headlining in the past few years. So it's extremely important to provide that habitat for these pollinators as industrialization continues to destroy their habitat. And though these gardens are small, they provide more benefits than meet the eye when considering the ways in which you can use them to get the community involved with these projects, which allows people to foremost care, but then to also be more educated and aware of these problems our pollinators are facing and the implications of those issues. Participation in these gardens paired with the use of a flagship species such as the monarch butterfly can prove to be a mutualistic relationship for both humans and our pollinators, as you're spreading awareness and action. And this engagement, as far as the human benefit goes, means people are out in nature, which provides what I consider a tremendous impact on the welfare of the human psyche, along with our physicality. And this is something I feel fairly passionate about on a personal level, and have shared many conversations with Karen Bailey, which is the founder and CEO of Kentucky Wildlife Center, who is equally as passionate and regarding that subject. And as far as that impact goes regarding the pollinators, it's pretty obvious, I feel like. So Along with the design and propagation of those plants for the way station, I'll also be in charge of maintaining the habitats of caterpillars that we will soon receive and their subsequent emergence and release. But sadly, due to the timing of this internship and the presentation, I can't speak as much on that aspect of my experience. And so I guess just some final remarks and takeaways is that this work is not easy. The employees involved sacrifice a tremendous amount of time and effort, and as well as sleep, to ensure the well-being of these animals that are in their care. The work is very demanding and extremely meticulous. It's not a field that receives much funding either, so you know that they're not doing it for the money. And especially when you compare that to veterinary practices for domestic species, these creatures don't receive the kind of applause our more cuddly friends do. So it takes individuals who really care to dedicate their time and money to this effort. The work they do is rewarding, though, from an intrinsic perspective. They love what they do, and they really see the value of these individual lives and the purpose they serve in our local ecosystem. And I think my biggest takeaway is something that was also prominent in my decision to choose this major, and well, to choose a environmental degree in general. And along with, some more confirmation hearing about this insane list of job opportunities and experiences that faculty have spoken out in regard to their career. But that is that you can attain a position in your adult life that, doesn't have to be an image of you sitting day to day in some soul-sucking position at a desk for your whole existence. You can have a job doing something you thought was cool as a child. You can have many jobs or experiences in this field if you so desire. And though wildlife isn't my final stop, I can add it to my own personal list of amazing experiences I will have had in my life in relation to this degree I'm about to obtain. And any questions?
Well, I'm going to jump in, Shay. This is Mary Arthur. I loved your presentation. I just thought you did a nice job of, really leading us along in what it looked like, what you did and what you got to work on and your excitement about it. And so I had kind of a question, because you did do such a good job and it looked like a really great internship. You said you were pleasantly surprised by how good the internship was. And that sort of made me wonder why, or, what you didn't think was going to be great about it.
Yeah, so I don't know. Wildlife isn't, so I'm a very big entomology nerd. And so that's why I kind of went for the pollinator part of it, which is, I'm really sad that I can't speak more about that, just because we're not at the stage that we're hatching, or well, I guess what we do is we're going to receive the caterpillar larva and then, provide habitat and watch, release and keep them, whatever. But so wildlife isn't something that I really care that much about. And I thought it was more mammalian focused. And like, as a kid, you think animals are so cool. And I still do think that, but definitely wasn't what I planned to do. But now that I see everything that goes into it, and I've learned so much about what it takes to do, and what these creatures contribute to our environment, it's really opened my eyes. And I think a lot of people kind of overlook this kind of position. And I mean, obviously they do. You have very few people that are involved in this, which maybe there would be more if it was funded better. But I don't know. That's probably not a great answer. But yeah, it's not something I was just initially interested in. So it's awesome what they do. And I've really seen some value placed on that aspect of environmental science.
Yeah, thank you for that answer. I think that's really, such a great articulation of your openness to the experience. And then also that you were able to sort of turn it a little bit toward the pollinator work, I think just speaks volumes of what you brought to it. So nice job, Shay.
Thank you.
All right, wonderful job, Shay. Thank you for sharing that with us. I am a particular fan of possums, so I appreciated the photos. Our next presenter is Rebecca Caldback. Rebecca worked at the UK Plant Pathology Lab. Rebecca, can you share your screen with us, please?
Can everybody see that?
Mm-hmm.
Okay. All right, thank you for that introduction, Dr. Sovey. My name is Rebecca, and I'm a double major in NRES and Ag Biotech. And this semester, I've had the opportunity to focus my efforts on a research project, and it's entitled Feeding a Growing Population. And the project focused on looking at the feasibility of applying leg hemoglobin protein as a visual marker for genetic improvement in crops. So we're all well aware that the global population is rapidly expanding, which has intensified food production and led to the decline of ecosystem health. Depletion of soil nutrients, fresh water, and biodiversity are all indicators of this issue. The productive capacity of land will continue to decline unless land usage becomes more sustainable. So this project focused on the fact that it's becoming increasingly relevant to locate sustainable and affordable sources of high nutritional value food, specifically in protein and fatty acids. So one crop of interest is glycine max, which is commonly known as soybeans. And soybeans are a very versatile crop with a wide-ranging habitat, which makes them readily available food source for people all around the globe. And besides their abundance, this nitrogen-fixing crop is also one of the most economical sources of protein on the planet. The plant contains soy-like hemoglobin protein in its root nodules, and like hemoglobin is responsible for the red pigment in nodules as shown in figure 5, which is over here. Like hemoglobin cooperates with nitrogen-fixing bacteria in the roots of legumes, and its protein is related to hemoglobin in the human blood, so it's responsible for the red color and the meaty taste of plant-based burgers such as Impossible Burgers. The goal of this project was to increase levels of omega-3 fatty acid and protein content by using like hemoglobin as a visual marker. If successful, the project could be environmentally beneficial in improving the current production of protein. Production of like hemoglobin-based protein is already using 95% less land, 74% less water, and producing 87% less greenhouse gases than conventional meat production. Besides the environmental benefit, it's also very valuable for human health. So it would provide an added health benefit to consumers. And outside of soybeans, this visual marker could potentially be applied to many other crops. The project has the potential to have far-reaching impacts on the food, health, and agricultural industries. So to perform this experiment, we extracted RNA from the root nodules of soybeans, as shown there in figure 5, and we made sure we selected root nodules that were pink to red, which indicates a healthy root nodule. The RNA was extracted from samples, and then we used that to synthesize cDNA. The cDNA was amplified using PCR, and then we did a bunch of agarose gel analysis to make sure that the like hemoglobin was present. We did some overnight cultures and purified the results of that, and then restriction digestion was performed and verified using another agarose gel. The PCR product and the vector underwent ligation, and the ligation products were transformed into E. coli bacterial cells using heat shock, and then plated to grow colonies. And COVID-19 impacted my research timeline like everybody else, so there are more steps to follow, but this is the point that I've reached so far. So for the results, the like hemoglobin was collected from the nodules and successfully amplified using PCR, and like hemoglobin protein has a known length of 464 base pairs. So if you look here in figure 2, you'll see that nodule 1 and nodule 2 both displayed bands between 400 and 500 base pairs on the gel, indicating the presence of like hemoglobin. So the vector and the amplified products in figure 1 were digested using restriction enzymes and displayed expected results, including linearization. Figure 3 up here displays the process of linearization using restriction enzymes. So restriction enzymes basically act as scissors to cut in a specific place, and you'll see the vector begins as a circle, and once you use these enzyme scissors, it became linearized. So that's why you see like a straight line here in the vector. Once both components are linearized, the vector and the protein product were ligated. So we have both sides and they're ligated here. And ligation basically refers to gluing the pieces together using the ligase enzyme. And this figure over here shows a more in-depth view of ligation, where you have sticky ends going together, forming a recombinant DNA. So after ligation, we formed a recombinant DNA, and then this ligated product underwent transformation into bacterial E. coli cells to increase our total product. Transformation occurred by the heat shock process, which refers to applying heat to the E. coli cells, and it basically stuns the E. coli cells enough that the nodule PCR product can attach to form a recombinant DNA plasmid. So basically you just kind of stress the E. coli cells, and then the recombinant DNA is able to enter the E. coli cell. And then we use E. coli cells machinery to produce more of the protein. The product was then spread on a plate, and I'm now waiting for colonies to grow before proceeding. And due to COVID, no substantial conclusion on the feasibility can be made yet, but so far the experiment has produced expected results in terms of gel results and transformation. And overall, it's been a fantastic learning experience, and I hope to continue this research in the future. And I would like to acknowledge my PI, Dr. Cashrew, and all the others who have aided in my academic efforts thus far. Does anybody have any questions?
Okay, I have a quick question. First of all, nice job. And I just have a question regarding these results you're waiting to hear or waiting to see that you're delayed on. I mean, how do you think they'll correspond to the gel results?
I'm hoping everything is running smoothly right now. So as I was mentioning, we were doing transformation and we got those to hopefully be expressed in the E. coli cells. So I spread it out on a plate the other day and waiting for those colonies to grow. And
Okay.
hopefully we'll see that the product was actually inserted successfully and is making our protein. And then the next step,
Gotcha.
express it.
Okay.
So, yep.
So Rebecca, are you expecting to finish this project before you graduate? And if not, do you have plans for finishing the project, and are there plans for publishing?
I'm hoping that I will be able to finish before I graduate in May, or at least find results that are satisfactory and enough to say, okay, the project is working so far, then I should continue the research. But basically after this next transformation step, if this doesn't work, then either I have to start that part again, or, then there's not really a point in continuing if it doesn't work after that step, or changing a different project. But yep. Hopefully the transformation does work though.
Well, now you have this whole crowd of people cheering for it.
Thank you.
That was really well done, Rebecca.
Thank you.
Yeah, Rebecca, I have one question. How did you get in touch with the plant pathology lab?
So I started working in research labs my freshman year, and I worked under Dr. Hildebrandt, and we focused on hemp and soybeans. So I kind of had some background experience with soybeans already. And this semester I transferred over to Dr. Cashrew's lab by a connection through Dr. Hildebrandt, because they were working on a collaborative project. So that's how I kind of got into the plant pathology lab. Mm-hmm.
All right, very, very nice. All right, our final speaker today is Michael Hagan. Michael worked with the, with the Chicago Animal Control. Right, all right. Michael, can you please share your screen?
I gotcha. All good? I think we're all good. Hi, I'm Michael. I'm a current NRES and business management student at University of Kentucky. This summer, summer and winter semester, I worked at animal control specialist in the Chicago area. Pretty much, you can tell by the name, it's animal control. Basically, we can identify what types of animals and pests are living near or around a customer's home, and then deal with the solution with long-term solutions and figure out the point of entry. This is located in Lake, Cook, and McHenry counties, and they've been doing it for now 30 plus years. So when I was working there, my role was pretty much centered around like the early stages of trap setup, basically learning what traps are better for which situation, proper baiting and methods, and just like where the location would be best, and then conducting repairs to damaged homes. One of the biggest things that attracted to me to this company, obviously COVID limited a lot of jobs that were out at the time. So what kind of drew me to this was their mission statement. And it was specializing in humane solutions to nuisance urban wildlife problems. And obviously as NRES students, humane solutions is a pretty big deal to us and a pretty big deal to me. So that kind of drew me to the company in general. So one of the first things I was really responsible for was damages and repairs. Zoom in here. These are just 2 squirrel holes in attics, and this is what I dealt with a lot of the days. What we do for this is we put a duct tape over it. Very, very experienced stuff, duct tape, but it's a really good indicator of when squirrels are coming in and out of the home. And what we do is we put a trap right outside of it, and we would know whether the squirrel was still living inside the house or not. And then to the right here, a little harder to see, there's a little hole there where a skunk is digging in underneath the porch. This is what I dealt with a lot as well, and I'll explain later how we dealt with that problem. Those are just some of the damages. I didn't really work with any coyotes or beavers. I set up one trap, which I'll talk about later. But those are really the damages I worked with on a daily basis. So what we do is we patch the hole using a cut sheet of a piece of sheet metal, and we spray paint it to the color of the house, obviously, and then caulk it so nothing else can get in. One of the things that I really enjoyed about this company was the fact that they cared about long-term solutions. There was a lot of other companies that they catch the animal, release it, and the house would have the same problem in the next week. It's really important to me that long-term solutions, and I think that promotes a healthier ecosystem as a whole in urban settings. Another frequent repair I did was on air ducts and different chimneys. We'd make screens for them. This is a box screen for an air duct. Raccoons often get in air ducts and go into attics. So we put box screens around them. Really easy setup. We just drill them into the roof. And then same thing for chimneys. We just bend the metal around the chimney and drill it in. The toughest repair I did while working at animal control were these skunk digs, where we put dig defense in underneath porches and decks. It would require a 2 foot by 1 foot trench all the way around the porch. And if you've ever dug into anything, you'll immediately find every bit of rock, electrical wires, sprinkler lines. So digging was definitely a lot of my job. And we put these screens up so that skunks can no longer get underneath, and then obviously put the dirt back over. The biggest reason why I worked at animal control was for hands-on experience with wildlife. I wanted to make sure that was on my resume as a wildlife management focus. So that was an awesome opportunity. I got animal control every day. I worked with animals hands-on. Those are the ones that they technically work with. While I was there, I only worked with raccoons, squirrels, skunks, bats, beavers, flying squirrels, bees, wasps, and hornets. Here are a few examples of the wildlife that I actually got set traps for and caught. Here are some baby raccoons that I hand caught that were in someone's wall. And then as well underneath is a beaver. We used a foothold trap for him. My picture is not as great for the skunk for some obvious reasons. I couldn't get as close to him. But we used this just a similar to this hole up top. We just put some boards around and make sure that he goes right into the trap. And then a skunk on top of a roof that got into a duct. Just in summary, this job experience was really unique. I don't think a lot of people have gotten a job opportunity like this. It was very interesting. It definitely is not what I want to do in the long run of my career. It was much more about human management than it was wildlife management. But it definitely was very, very interesting. I got a lot of really good connections from it. So I'd recommend it to anybody. Thank you very much. If anybody has any questions, please let me know.
Hey, Michael, I'll ask a question. This is Steve Price.
How are we doing?
Hey, so good. How are you? Good job on your presentation. I was just wondering how some of your coursework, especially wildlife-related coursework, helped you out with this internship opportunity.
Mammalogy definitely helped in a lot of different ways. One of my favorite parts of the job was when we go to a house without any idea what the animal was, and it was kind of like being a detective. And mammalogy definitely helped with tracks, different skeletal features, and just allowed me to be a lot more ahead of my game when it came to identifying what animals could be the problem, where they're entering, and different things like that. So definitely mammalogy. And then later on, a little bit of ecology and forest ecology really helped because in Illinois, we have to euthanize all raccoons and skunk. That's under the wildlife code. But as well, they're euthanizing beavers. And something I actually taught my boss while I was there is how important beavers are as keystone species to the environment. So now I'm helping them look at where we can relocate them after catching them. So definitely a few classes that helped out.
I'm curious, what are the options for relocating them? Is that kind of taking them to a natural area or?
Yeah, it's definitely tough. That's one of the issues that they have right now. And and moving forward into just being more conscious, humans, is tough for them, especially in Illinois. There's a lot of different rules around it. They have to release 10 to 40 miles away from where they caught it. And there's a lot of animals that they can't be released. But for bees, wasps, and hornets, they have a bunch of nests out back where they can put them. And then as well, for beavers, we brought them to the Illinois Wildlife Center, similar to what Shay did. Not to put that on them, but it was just much more hard, much more up their alley to find a place for them.
Yeah, we do have a lot of people bringing or a lot of animal control. There's a lot of public that kind of come and bring stuff, but we do get a lot of intakes from animal control. So I was going to ask a question about that, but you answered it.
Yeah, no, it's definitely like something that when I got there is the reason why I don't really want to do it in the future, is learning everything about wildlife management and then kind of doing the opposite. So this is not what I'm doing in the future, but it was interesting seeing it from that side. And it was definitely one of those things where they, like few of my colleagues that I was working with, just didn't know the true impact some of these species have on the ecosystem around them. So it was a learning moment for them as well.
All right, wonderful. Thank you so much for sharing with us, Michael. All right. So that brings us to the end of our internship forum. I guess we're a little bit close, so we have about 5 minutes left till 6:30. So I'm just gonna open the floor up for any of the participants to ask questions of their peers, or for the experience advisors to ask questions of the presenting group. So you can feel free just to unmute yourself and ask.
I have a question I'm just going to throw out to all the students that presented. What would your ideal internship have been if it wasn't a pandemic, or did you get the internship that you wanted?
I'll speak first on that. I would have loved to do stream restoration or just stream assessment with like benthic macroinvertebrates. I'm very interested in those. So I do plan to apply for a couple of positions relating to that later, but yeah, it wasn't possible for me during COVID.
I definitely wanted to do actual water sampling of Harrington Lake, and we wanted to do lab work, but just because of COVID restrictions, like Ben and I weren't able to like get together, and then also the lab wasn't welcoming samples, so we couldn't do that. So we just had to use like data from that was already recorded.
I was supposed to go up to Philadelphia and do some environmental consulting with an internship that I got up there this past summer. So ideally, I would have liked to have been doing that instead. And that basically just involved water quality monitoring, air quality monitoring of factories, and just making sure that they're staying within standards, and also dipping my toes into some industrial health work as well. But now it kind of worked out well, because now I'm going to get to go do it after I graduate, and hopefully it would lead right into a job. So now it can be one more consecutive experience rather than breaking it up. So I guess it worked out.
Yeah, before COVID. Oh, go for it, Rebecca.
I was supposed to be doing a internship with USDA, so I missed out on that last summer, but I was happy with the experience that I got this semester working in the lab. Got a lot of hands-on experience and good connections with some more professors. I obviously wish that my project was further along because of COVID and restrictions on getting back into the lab. That was a bit delayed, but overall it was a great experience. So
yeah, I had a internship lined up with Kentucky Fish and Wildlife, tracking elk out in Hazard, Kentucky, and elk management and stuff like that. And then COVID ended up finishing that.
Yeah, and I was supposed to be in Paducah doing environmental remediation at the old nuclear plant down there. But it worked out well for me, because I've actually really enjoyed the stuff that I ended up doing, because I don't think I would have gotten the opportunity to participate in research had COVID not happened. So kind of grateful for that in a weird way.
Yeah, absolutely.
All right, let's.
Absolutely.
All right, let's. 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 All
Thank you for that wonderful question, Sandra, and for all of the presenters' responses and answers to that. If anything, it shows just how talented and resilient you all are to, so quickly change speeds and still get so much out of your internships. I think it also says a lot to your to the credit of your supervisors and mentors who are able to also help you navigate that. So a big thank you to all of the mentors and supervisors that joined us, and thank you so much for our students for presenting today. And we are out of time. So have a wonderful evening, and I'll see you all soon, hopefully.
Yeah, great job, everyone.
Thank you all.
Thank you all. Nice job.